Okay, so today's flipped lecture was on operons. What was, I'm going to ask this every time, what was like the basic theme?
Operon.
Yeah, but like beyond that, like explain that.
So, I mean, basically operons are bacterial sequences controlled by
Genes, they're bacterial genes.
Yeah, genes controlled by one promoter.
Yeah, so really a big focus on this is two parts. It's a continuation from the last lecture where we talk about the structure of bacterial genes. But this was a lot of heavy lingo and a lot of heavy, information more about transcription. So again, like the regulation of the genetic circuits, how do you actually, if you have a gene, if you have this operon, how does it get turned on? How does it get shut off? How is it regulated? What's controlling transcription? And I went through like a giant list of all these things that add to the complexity of the regulation of transcription. That was essentially like the gist. And then I went through, okay, so first before I go to the next point, let's recall kind of like the two most important things from this lecture. One was I defined what an operon is, and I gave you like 4 things that operons sort of like have. What are those 4 things?
Synteny.
Yes, so synteny, which was what? The order of genes. So it's almost like if you were imagining each gene is kind of like a sentence. It has kind of like a meaning and a function. And you could imagine synteny would be like a paragraph where you're stringing genes together in a right order, and that order matters and it has a meaning and it's important. So synteny is the order of genes. And sometimes that order can get like whacked if there's some kind of like a recombination or something. The order can evolve. So, but synteny, operons, in operons, synteny is usually conserved, which means it usually stays the same. It's selected for because it has a functional like meaning, if that makes sense. Okay, what else was there about operons? Co-regulation. What does that actually mean? Explain that.
It's multiple, right? You can regulate multiple things at once.
Yes. So usually there might only be like one promoter in an operon that regulates maybe 2 or 3 or 4 or 5 or 6 genes. So it's kind of like, again, the bacterial genomes are always trying to streamline. They're selected to be smaller in content. So if they can sort of, in computer language, they call this bum the code, where you get rid of sort of extraneous sequences and get the same functionality out of less coding, that's beneficial for them. So oftentimes they might put like one promoter in front of like multiple genes, and that would be co-regulation. They're regulated together. What else? The 2 other things?
Polycistronic.
Polycistronic transcription, and that is what?
Where one sequence or one frame encodes for multiple proteins.
Not necessarily one frame. When you talk about frames, you're talking about the codons in translation, so that's a step ahead. But I think you have it right in your mind, but you're not saying it right. Polycistronic, a cistron is a messenger RNA molecule. So polycistronic means one gene. It's not, central dogma kind of teaches you one gene makes one messenger RNA, makes one protein. That's not really true. Multiple genes can make one messenger RNA. That is the length of both the genes. And then this can get translated into 2 proteins. So polycistronic transcription means there's more than one cistron in this transcript. There's 2 cistrons, 1 and 2, which get translated into 2 different proteins. And this is often important for viruses. So like flaviviruses, like I said, they have like 10 genes. Sometimes they'll literally just have one long line of genes, and it will make one long messenger RNA, which depending on the virus might get translated into one giant protein and then chopped up into little proteins, or it might be translated into separate proteins. But this is an example of polycistronic transcription. So one messenger RNA might code for a couple genes. And what's the final thing? Shared function. So I gave you the 3 examples. In addition to those 3 examples, what you might often see is like ribosomal genes. So the ribosome is a giant complex of proteins and RNA. And oftentimes you'll see ribosomal operons that might have one promoter. And so a chunk of the ribosome gets made here, and here, and here, and here. And then these things come together and form a complex, a ribosome. So that's just one extra example. And the 3 that I gave, we're going to go over now, again, because these are the ones you need to like memorize. So this will for sure be on the test. I always put this on the test, just so you know. Like, might even be on the next quiz. I tell you so that you don't get it wrong. Like last time I said, draw glycine. If anybody gets this wrong, I'm going to be super upset, because I'll just be like, okay, you're not even like trying. You're literally not even trying, okay? That's my test to determine who's trying. Okay, so now so let me also first ask, since we kind of know the gist of the lecture, were there any questions about it before I go into the 3 operons?
Can you go over IRES and T2A?
IRES and T2A. These are like hacks. These are hacks to make like pseudo, this is not a real word, I'm making it up, pseudo operons. So eukaryotes, do eukaryotes have operons? No, but imagine here's a good scenario. This is actually like a really good for the flipped class. I'm really glad you asked that. And we actually talk about this in the future in the, there's a lecture called psilocybin biosynthesis. Psilocybin is the drug in magic mushrooms that makes you hallucinate. We actually talk about how to recapitulate the psilocybin biosynthesis pathway. There's 4 genes that create psilocybin, okay? So imagine you want to make an organism that makes psilocybin. And in that lecture, I give the example of making magic beans. So imagine you wanted to grow like sweet peas or beans that make psilocybin. Okay, you would have to clone out the 4 genes that create psilocybin, and you'd have to insert them somehow. Okay, this can sometimes be a problem, because imagine these are eukaryotic genes. So each of these genes in this organism is not organized as an operon. Each of these genes has its own promoter, its own terminator, and you can imagine it's this times 4. How long do you think this is? Let's imagine this gene is 1,500 base pairs. Let's imagine a terminator is 1,500 base pairs. Imagine a promoter is 1,500 base pairs. That's now 1,500 times 3, 4,500 base pairs total for 1. So times this by 4. I don't know what the answer is, but let's see. Imagine it's 5,000. It'd be 20,000. It's about 20,000 base pairs. How big is the biggest plasmid? Your topoplasmids that you work with, how big are they? You should know that. They're probably about 3,000 base pairs. Even if you insert a gene, maybe it's like 4,500, 5,000 base pairs. You cannot work with plasmids this big, okay? So remember, I talked about the different sizes of things. That actually has a purpose. When you start building stuff, you can't just like imagine you wanted to write a book, but the page limit you were given was like 3 pages. Okay, you can't do that. So like if you're gonna clone these 4 psilocybin biosynthesis pathway genes, you don't have space for 20,000 base pairs, because you can't put that into a plasmid. I mean, there are ways, hacks we'll get into in the future, but for now, like you can't put that into a plasmid. You can't easily insert 20,000 base pairs into a transgenic organism. It's just too big. So you have to figure out a way to get rid of a lot of this extra sequence. Okay, so what can we get rid of? Well, if we make like a fake operon for a eukaryote, we could get rid of the terminator, we could get rid of 3 of the promoters, and maybe we just have 1 promoter. Okay, so imagine we wanted to take the 4 genes and bum the code, get rid of the terminators, get rid of the promoters, and just have the genes. We could make a fake operon for eukaryotes. So we could make a genetic construct that looked like this, where it had 1 promoter that was turning on all 4 at the same time, because we need all 4 to make psilocybin, okay? You turn on all 4 at the same time, now there's a problem that we have to fix, which is this is going to be 1 polycystron. How does a eukaryote know how to read this? Does a eukaryote know how to read polycystrons?
No.
Not really, okay, because they're not evolved for it. Prokaryotes, this would work right away. But a eukaryote, so like the beans, sweet peas, whatever the species is, they're not going to be able to read this. So in order for this to work, there are these sequences that you can insert into this to sort of like hack the code and make it work. So a T2A is a sequence from a virus, okay? And if you put T2A in between each of your genes, and T2A is pretty short. Maybe it's like 100 base pairs. Okay, so it's not very costly. You put a T2A in between each of your base pairs, and what it does is it's a trick that a virus has evolved. It's a special sequence that when the ribosome is reading this transcript, when it sees a sequence that was coded for by the T2A, so the T2A is a DNA sequence that gets put into messenger RNA, and as the ribosome is reading that messenger RNA and it's putting those codons into order, when it hits the T2A, it hits a special sequence, which is PGP at the end of the T2A, proline, glycine, proline, and as soon as it hits this, the sequence before this causes the ribosome to malfunction, and it skips a peptide bond, so it jumps. It essentially just jumps. It doesn't make a peptide bond. And so if it doesn't make a peptide bond here, 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 and here, now you've produced 1, 2, 3, 4 separate proteins via this little virus hack. Does that make sense? That's an advanced concept, but does that make sense?
So it's essentially like an apostrophe of genetics.
Yes, kind of, yes, like exactly. Like, and that's literally what this class is about, teaching you, like, okay, how do you like hack the code to get what you want? And again, this solves a problem for a biotechnologist. The problem is you don't have space to put in 4 terminators and 4 promoters. You just don't have that space on a typical plasmid. So you have to cut that code out and figure out a way that you can turn on all these genes at the same time when you turn on this 1 promoter. And so you can do that with the T2A. The IRES is the same thing, but a different concept. Or same concept, different thing. T2A is from a virus, okay? And IRES is an internal ribosomal entry site. So if you put an IRES at each of these points where the T2A was inserted, now what happens is something a little bit different. A ribosome will come in, and one will start here, another ribosome will come in and start here, another ribosome will come in and start here, and another ribosome will come and start here, because it has an internal ribosomal entry site. Does that make sense? So these are two, again, these are like advanced concepts, but these are 2 genetic sequences that you can use to make like fake operons in eukaryotes. Okay? Good question. So do you understand what they are and why you would use them? Yes?
Yeah.
Okay. Okay, the 3 operons. Let's start with the simplest. So now you're actually learning biotechnology. Most of the plasmids that you will ever work with for the rest of your life use the lac operon. They're like the most common plasmids. A lot of the plasmids use the era operon, and a lot of the plasmids use the trip operon. So now I'm teaching you like biotech stuff that you're going to use. Okay, so lac operon. So Lac operon is the simplest. There's a few genes. Let's make an operon. I think there's 4. You can fact check me if I'm wrong. And then there's one outside. So the lac L, what is the lac L? This is the start of the operon. And this is a gene that's outside of the operon but regulates it. This is a common pattern you're going to see in operons. Is usually there's an operon that does something, and then right outside the operon is something that regulates that operon. Okay, so what is lac L? It's a repressor. What does that mean? It means it's a transcription factor. It also means it's a special type of transcription factor. It means it's an off transcription factor. If it's bound to the promoter, it's off. Okay. What does the lac operon do?
Creates lactose.
It digests lactose. It doesn't synthesize lactose. So this is metabolism breaking things apart. So it finds lactose. One of these is an importer. I think it's this one. Maybe it's lac A. Lac A finds it's an importer, which means it's a protein that gets put on the surface, and if it sees lactose in the environment, it shoots it into the cell, transports it in. Okay, one of these is lac Z, which is beta-galactosidase. That's what actually digests the lactose. And I think there's some other ones. Oh, it's just 3.
Lac Y.
Lac Y. Maybe lac Y is the transporter. It doesn't matter. You don't need to memorize these. You want to know lac Z, and most importantly, you want to know the repressor, because this is what your plasmids will have. Okay, so if the repressor is red, repressor will bind the promoter, and now what's the state of the gene?
Deactivate.
It's off. Okay, now you add lactose, and what happens? It turns on. How does it turn on? How?
Kicks out.
It what?
Kicks lac L off.
Yeah, well, lac L essentially binds to the, this would be called the ligand, something that it binds to. It binds to the ligand. There's a conformational change. Remember, proteins are these folded structures, and if you all of a sudden have an interaction, you're going to get a change in that fold, okay? So as soon as it interacts, it changes its three-dimensional structure. Now it's no longer fit to bind the DNA, so it falls off. Now it's like over here. And now what's the state of the gene? It's on. And so now what's happening? Genes that synthesize the destruction of lactose are now being made. Now lactose is being eaten. As soon as lactose is gone, what's going to happen? You eat all the lactose, what happens?
Lac L rebinds.
You eat this. Now this thing rebinds and shuts it off. Like, do you see how important this is? This is literally just like turn the lights off when you leave, like saving energy. It's literally like a switch. If lactose is there, you turn the genes on, you make the stuff that can eat it, and then as soon as it's gone, you shut it off so you're not wasting energy. Okay? So all the rest of the operons are essentially like this, but like derivatives, or a little bit more complexity. Okay, so the ERA operon, this is the one I use the most in the lab. Oh, actually, before I go to the ERA operon, what's IPTG? This will be on the test for sure.
It's a lactose mimic.
Yes, IPTG is so what you actually use in the lab is you don't use milk sugar, you use IPTG. Why do you use IPTG? It's a lactose mimic. Why do you use it? Yeah, because the cells can't digest it. So usually when we want to turn genes on, we usually don't want them shutting off. We're usually like, okay, time to make the protein, turn it on, and we want it on, and we don't want it shut off again. So you use IPTG in the cases where you don't want it to shut off, which is most of the time. Okay, ERA operon. What do you think ERA operon does? What do you think operon does?
Metabolize arabinose.
Yes, arabinose is a sugar. The operon genes are responsible for eating arabinose. Okay. And just like I said before, this one's a little bit more complex. There is like, I'm going to erase this so I have room. There's like a couple regulatory things, and then there's what is the transcription? It's RNAseq. RNAseq. RNAseq is what of the operon? It's the regulator. Is it we call it a repressor? It's kind of a little bit more complex than just a repressor. Repressor works for a lac operon because it's a very, very simple, like it binds, it's off. This is like a little bit more complex. So I would call it like the regulator. I don't actually know like what the actual precise term for it is. It's a little bit more complex. So RNAseq gets made, okay? And in the picture I show you, RNAseq is like this little like protein that looks like this. And when RNAseq is made and there's no arabinose, let's have black will be the DNA. The DNA essentially folds and creates, like, you could call this like a stem loop. Okay? So the DNA folds, if this is on to the rest of the genome. When the RNAseq protein is bound, there's a stem loop in front of the promoter. And so now what's happening? Is it on or off? It's off. Okay? Now you add arabinose, which will be like this red triangle. You add arabinose, that binds to the regulator. It binds to the regulator, there's a conformational change that releases the stem loop. Stem loop is no longer there. This thing switches over a little bit, so it like shifts to this new section. The key point is it releases the stem loop. So now what's happening to the genes? Are they on or off? They're on. Okay, so it's a regulator that induces a stem loop or no stem loop based on whether or not the sugar's there. So if you're getting the gist is these genetic circuits are all, you add something plus X and something happens to the genes. They shut on and off. And it's all based on whatever that X is binding to and whatever the ligand is binding to is a transcription factor. Okay.
Why did they name the regulatory gene the same as one of the structural genes?
Did they?
Yeah. So you have RNAseq.
Oh, it is. I bet it was discovered after.
Okay.
So this is that's an interesting question. Usually when genes are discovered, that's usually when they get their names. So I bet the first one that was discovered was A. They probably figured out it does something with arabinose. Then they would have discovered B and C. And so they would have said, this is the operon. It's the operon, A, B, and C. And then somebody later probably discovered like the regulator of it. Or maybe it was discovered at the same time and they both named it C. I have no idea. That's an interesting question. But terrible naming, right? Bad idea. Yeah, weird. I bet now there's kind of like something that distinguishes them. It might only be in that figure might be old, but that's a really good question. Good detail-oriented thinking. Names are important. Okay, the hardest one. I always get actually kind of terrified when I have to teach this one. Oh, because it's even hard for me. The trip operon. But the reason I continue to teach it is because I think doing difficult things is important. And I also I have some plasmids in the lab that use this. I've never used them, but I have some plasmids. I own some plasmids that do this. So I feel like I should know what it does. Okay, I'm going to try to synthesize it in my own terms, okay? And so this should help. Simplify and point out the things you need to remember, the things that are just extra crap. Okay, so the trip operon, imagine some operon, let's just say X, Y, Z, okay? These genes, what are they going to be important for? Yes, the synthesis of tryptophan, which is what? What is tryptophan? It's an amino acid. So do you see how like knowing your amino acids would be important? Now you can understand the purpose of the tryptophan operon because you know your amino acids. Okay. You have to synthesize tryptophan. Actually, do we have a tryptophan synthesis pathway? I don't think we do. Yeah, I think tryptophan is one of the amino acids you have to get from eating. And you eat it, like turkeys have a lot of it. I don't, fact check me on that, but I actually don't think that we synthesize tryptophan. So some of the amino acids we don't even have synthesis pathways for. We have to get them in our diets. But okay, this is the operon that synthesizes tryptophan. Okay, so what do you think is going to be the ligand that regulates the tryptophan operon? What would be the ligand? So in the lac operon, the ligand was lactose. In the air operon, the ligand was arabinose. What is the ligand in the trip operon? Tryptophan, the amino acid. Okay, so tryptophan will be the ligand. Let's just make it a red circle. Okay, now we can also logically think this through, okay? If we have a lot of tryptophan in the cell, do we need to be synthesizing more of it? Think that through again. If we have a lot of tryptophan in the cell, do we need to be making more of it?
Well, this is the one that titrates, right?
Yes, it does. But what I'm asking is like, imagine, okay, if you've already eaten 20 donuts, do you need another donut?
No.
So I'm saying like, if you have like a lot of tryptophan in the cell, do you need more of it? No. Okay, so I'm telling you the logic of the circuitry. The logic of the circuitry is if you have a lot of tryptophan, so a lot of tryptophan, then it's off. Because it's like, we don't need any more, we have a lot. Okay, if you have a low amount of tryptophan, it turns on. Okay, so this is like the basic understanding of the basic circuitry. Now it gets a little bit more complicated. Okay, a couple of things you need to know are side points. As messenger RNA is being made, at the same time, literally at the same time as it spits off the RNA polymerase, it's also being translated. So, and this is not always true, okay? So biology is not black and white. Again, like this is not always true. But in many cases, as messenger RNA is being transcribed from the RNA polymerase, as it's popping off the RNA polymerase on the back end, a ribosome is sitting there ready to translate it. Okay, this is what's happening in this scenario. Okay, so let me give a little bit more space for the promoter. Okay, and this first gene. Okay, so as this thing is being synthesized, let's synthesize a long transcript, which is this. And let's give this one a name, let's call it 1. 1XYZ, okay? So here's 1XYZ in the transcript. Okay. So the RNA polymerase is here making this transcript. It's reading this DNA, making this transcript. Label it RNA pol. Ribosome is already on the end making some stuff, making some protein. Okay, so there's a special gene, and the name of the sequence that's important is the attenuator region. Attenuator region. Okay, in the attenuator region are a whole bunch of codons for tryptophan. What's the symbol for tryptophan? Is it W? Fact check that. I thought it was what's Y. Wait, what's Y then? Because now you got to correct my memory. What's Y?
Tyrosine.
Tyrosine. Okay, so I confused tyrosine and tryptophan. Okay, good, very good, very good. Okay, WWF. No, it's WWWW. So there's a sequence in these codons which the ribosome wants to read and translate into WWWWW. So what's going to happen in the situation where there is a lot of tryptophan?
you said it would turn off, right? Or too much, or?
Well, okay, this is the final logic. The whole circuit will turn off, but now I'm explaining the mechanism of how the circuitry actually like gets to that final shutoff. So if there's a lot of tryptophan in the cytoplasm, there's tRNA charged with tryptophan, ready to be loaded into the ribosome, what's going to happen when it sees this www? Will it be easy or hard? It'll be easy. If there's a lot of tryptophan in, okay, this is involving like circular logic, so be careful, think about this. But if there's a lot of tryptophan, okay, there's a lot of it in the cytoplasm, it's ready to get thrown into the ribosome. The ribosome's going to go super fast. It's just going to blitz through this region because it's there, it's ready to go. So it's going to blitz through, and it actually goes so fast that it pops off the RNA polymerase. So it goes so fast that it catches up with the RNA polymerase and it knocks it off. So now it's no longer being translated. Okay? You're not making these proteins. The transcript, the polycistronic transcript is present, but you're no longer making these proteins because it popped off the RNA. Wait, sorry, I said that wrong. It pops off the RNA polymerase, so it stops making messenger RNA. Does that make sense? I kind of botched that explanation, but I hope that makes sense. Let's do the opposite, unless you have a question.
So, I mean, it's kind of similar as far as the mechanism of Rho.
Yeah, except it's like opposite. Rho is like a protein that catches up with and knocks the ribosome off, or is it the RNA polymerase? No, it is. Yeah, it's similar. Rho is Rho's termination, right? So it's transcription. So a Rho-dependent termination would be catching up to the RNA polymerase and knocking it off. Yes. So do you see how hard it is to think through all these languages very, very fast? Yes, but you're right. It's very similar to Rho. It just catches up and knocks it off. Now, in the situation where there's no tryptophan, we need it. We need it to be making all these proteins because there's nothing there. There's no tryptophan there. So the ribosome slows down. Okay, it slows down when it hits this spot because it needs tryptophan, but there's none around. So it slows down when it's trying to code for or translate for this www. It slows down. That allows the RNA polymerase to finish the transcript, and then the transcript is present and it gets translated. Okay? So this is really complicated. This shows you some of the complexity that you can have in these genetic circuits. And this is actually like an analog circuit. So a digital circuit might be like on or off. Like lac operon is kind of like on or off. It's like binary, yes or no. This is analog in a sense that like it can actually like if you have like 3 tryptophans, maybe it will do like 3 times the synthesis. Or if you have 1 tryptophan, it would do 6 times the synthesis. Like it actually like adjusts to how much you need.
If you had no tryptophan, would it not work?
If you had no tryptophan, then it would be like, if you had 0 tryptophan, then it would be on at like its highest level.
But could the ribosome go any farther? Would it get stuck at the different?
Wow, that's a good, well, so what, I see. I don't have the answer to that question. That's a really, really good question. I want to like write that down because I have to figure that out. What happens if there's none? Again, I hope I didn't botch the explanation for this because it's super complex. I usually don't put this on the test, but I try to like walk through it because again, I want you to understand like how much complexity there can be in regulating these circuits. And this is a circuit that's used in biotechnology. I'll try to answer this question before the next class. Thank you for asking that. Okay, I still got time. So those are like the main points from the thing. Let's try to dive a little bit deeper. How do you actually study transcription? How do we know these things? Like, how would you go about studying transcription? Let's first start thinking this through. Like, what molecule are you going to be looking at? if you're studying transcription,
Nucleic acid.
yes, which nucleic acid? Yeah, which nucleic acid? Which one?
DNA.
Well, okay, I mean, I guess this may be an unfair question. You'd probably be looking at both. But like, which one are you going to be measuring?
mRNA.
Yes, you're gonna have to figure out like schemes of measuring messenger RNA. Okay, so let's kind of talk through some of the basic, since you've seen the data structures, what are some of the basic ways in which we could measure messenger RNA? Think back to the previous lectures. What would be like the easiest way? How can you measure messenger RNA? Like, is it there? Is it not there? How do you see it? What would you use?
I mean, you could use Sanger sequencing.
Sanger sequencing is typically used for like determining what a PCR product is or what's in a clone construct that you build.
Okay.
You wouldn't use Sanger in the beginning. What would you use? What would you need to do before that?
Agarose gel.
It's closer. Like even before the agarose gel.
Oh no.
Like, okay, so we all agree that like we're gonna look at messenger RNA if you want to understand how transcription works. So like essentially, just to re-explain this, if you wanted to actually like study the lac operon, like when is this thing turned on? How does this thing turn on? You kind of want to look at, well, when is it on and when is it off? You need to be able to measure the transcript to determine when it's on and when it's off. You get that logic, right? Okay, so the first thing you're going to look at is messenger RNA. So let's maybe like start with purifying messenger RNA. Okay, so we got that. Then what do you do with it? You purify messenger RNA from a cell. Are you going to just have one transcript, or are you going to have like transcripts for every single thing that thing was making? What's the genome of E. coli? How big is it? How many orfs?
5,000.
There's 5,000 orfs in E. coli. How many do you think are on at any one time? It's not like one, right? Like it's a high number, right? Probably in the thousands. Okay, so let's imagine it's probably even higher than that. But let's imagine at any one point in the cell, there's 2,500 messenger RNA transcripts. How are you going to tell which, like, amongst those 2,500, how are you going to know if your lac operon is on? Like, what would you do? Think back to, like, measuring if COVID-19 is there.
Is it reverse transcriptase?
Yes, like the first thing you're going to have to do, you're going to have to find some strategy of PCR, because you're going to have to find some strategy to essentially like magnify the thing you're looking for. Okay, the thing you're looking for is one transcript, the lac operon, amongst 2,500. So how do you zoom in and look at it? Well, you're going to use PCR to amplify, to zoom in on nucleic acid sequences. You can't PCR, you can't just PCR. Why? Yeah, because you're measuring transcript. If you just did a PCR reaction for the lac operon, it would always be positive in the particular bacteria because your bacteria has those genes. Okay, so you can't just use PCR. You have to use RT-PCR. Okay, and remember RT-PCR had the first step of the cDNA synthesis. Okay, and that's going to synthesize. It's going to have messenger RNA with a complementary DNA. So the primer you would design would be a specific primer that targeted the lac operon sequence on the 3' end. Then you would do a reverse transcriptase reaction to synthesize the cDNA. And then you could PCR that. And then you could run an agarose gel. And you could determine on or off.
How would the primers just target the cDNA, not the regular DNA?
Oh, that's a good question. The answer to that is they don't, but there's a way that you get rid of the DNA. So this is an advanced question. Very, very good. Very smart. So the question is, After this, the second step is a PCR, okay? You're gonna have 2 primers, and in theory, it's gonna be the same one. In theory, you're gonna have a forward and reverse that recognize the quote-unquote cDNA. How do you discriminate cDNA from the original genome, which is gonna have the lac operon? The answer is you can't, but usually before 1 and 2, you add a step. You add a step, 1.5, which is called DNase. What is DNase? Anytime you see an A's, what is that in molecular biology? It's an enzyme. What do you think DNase does? It degrades DNA. So usually there's a step between 1 and 2 where you add DNase. DNase will kill all the genomic DNA, but your cDNA will remain. I don't know how it distinguishes between, I don't know how the DNase knows not to kill the cDNA. It might be because it's like a complex with RNA. But usually that step, that will get rid of genomic DNA, and then your primers are no longer targeting your genome. They're only hitting your cDNA. And usually there are a bunch of controls that you run with this. So you would run, probably be worth enumerating the controls you would need for a typical reverse transcriptase PCR reaction. There's typically your positive control, you have a positive control for DNA. So if you had 2 primers that targeted the lac operon, you would run a reaction with the genomic DNA, okay? So let's draw a giant gel here, and I'll show you all the controls you typically do for a reverse transcriptase. Let's say this product is 2,000 base pairs. So we run our ladder, okay? The first one, you're doing the PCR reaction. Let's say this is 2,000. Okay, so the first one, you run a reaction on the DNA. What's this proving? It's proving you have the gene there. Like at least the gene is there, so in theory you could make transcript. If this is off, it means either your PCR or your RT-PCR in the wrong thing, or like the gene's not there, or something's wrong with your primers. Okay, so this is like a positive control for like primer functionality. Okay, and then you'd usually do a minus RT and a plus RT. RT is reverse transcriptase. So you do a minus RT reaction on the original sample, which means you never added reverse transcriptase. So cDNA could never be made. This should be completely negative. So this is a control for if there's nothing there. There's a control for that you're not amplifying your genomic target. You have to carefully distinguish the words I'm saying. This is a control for not amplifying the genomic target, the DNA. Okay? The plus reverse transcriptase is your actual test for the cDNA. And if you see it looks like this, okay, then you're good. Then you say, if I don't add the reverse transcriptase, I'm not getting that, therefore it's not amplifying the genomic target. If I do add the reverse transcriptase, I'm getting the cDNA, and it's on. If you did this, and you didn't see it, let's say this is sample 1 and sample 2. If you did this, what would you conclude? And there's nothing there, what would you conclude? So you add the reverse transcriptase, it's finding or not finding the transcript. What would you conclude if there's nothing there?
Didn't do it.
It's not finding the transcript, so it was not on. So if your result looked like this, you would conclude it was off. If your result looked like this, you could conclude it was on. And you see you could do now a bunch of experiments where, well, what happens if you add lactose? You're going to see it. What happens if you remove lactose plus RT? It's going to look like this. This should be moved over a little bit more. So now if you do a bunch of these different experiments, you add lactose, it turns on. You remove lactose, it shuts off. Now you can conclude that lactose is the ligand regulating the operon. This would be like a start to how you could analyze the transcription of an operon. Does that make sense? Okay. 4 minutes. Any questions? I didn't even get through. That was like the first, that's like the first method. We don't even really do that anymore. Like that's like the most basic method. Now usually what people do, I'll skip the tiling arrays because we don't use them anymore. Now usually what people do is RNAseq. So remember how I told you if E. coli has 5,000 genes, let's guess, maybe 2,500 are on. Now what people do is they just make strategies to synthesize one, you extract messenger RNA. So you got all those 2,500 to make cDNA from all 2,500. Okay, so one way you can do that is you can have a bunch of random hexamer primers, which are random, so they're going to randomly hit a bunch of these, and if you have enough randomized targets, you hit them all. So you just make cDNA for all, okay, and then you can feed this into a sequencer. What kind of sequencer would we use? What?
Illumina?
Yeah, you would use a high throughput sequencer, so maybe Illumina. Right? That's the distinguisher between when you use Sanger and when you use a high throughput one is if you have a lot of stuff you're looking at, you usually use high throughput. So then you would sequence these with Illumina, and then you just get back a list of here's all the things that are being expressed, and here's their relative levels, the quantity at which they're expressed, and we would call this a specific name. What would we call this? A transcriptome. The book, a tome is like a book, the book of all transcripts that are being transcribed at that one point when you extract your messenger RNA. And then you could just look through, is your lac operon in that list? That's probably how you would do it today. And then you would do a bunch of different, like comparisons, treatments, make a bunch of different transcriptomes, and then compare the transcriptomes with computer algorithms. It's going to get you like a lot more data. But the other way is sort of a lot more personalized, if that makes sense.
Is it cheaper too?
It is cheaper if you know how to do it. So like the biggest expense in lab work is training and troubleshooting and paying people for time. That is the biggest expense. So there's like clearly a scenario where you're trying to teach an undergraduate reverse transcriptase PCR, and it could take you like a whole year, and it might just be cheaper to spend $5,000 and get like a transcriptome. Yes, but it depends. So it depends on like skill level. Like I could go in and I could do that, and I could get it to work. Get it to work within the first 3 times, and it would probably cost like $500, the cost of the kit. So it's definitely cheaper. And oftentimes you would do both. If you were studying like a very specific protein, you might literally do both so that you can make a stronger argument. And you say, it's being expressed at this time point in both the RT-PCR and the transcriptome. Oftentimes they do RT-PCR as a confirmation that the transcriptome worked. So again, what you see in papers is you see sort of like strings of these methods. I did this, it worked. I also did this validated my previous conclusion, and then I did it a third way and it validated my previous conclusion. So that's oftentimes like how people go about it. Okay, let's end this.